Hey, A&Pers, this is Dr. Jones checking in. This is audio-video lecture for A&P II Lecture Test 6. This is the genetics lecture, and I have no idea what number lecture we're on. Looks like this will be lecture 8. And hopefully, fingers crossed, our last lecture. If this gets too long and unwieldy, I'll tear it down and turn this into 2 lectures if we have to. So for our last lecture, we are talking about genetics, genes, chromosomes, and inheritance. In the note set, this is the bottom of page 22, section 29.9. So the first term we need is chromosomes. Chromosomes, there are 46 chromosomes in humans. They come in 23 pairs. So you've heard me say this before, but I use the example of 23 pairs of shoes. If you have 23 pairs of shoes, you have 46 individual shoes. In chromosomes, like shoes, the 2 shoes aren't identical because you have a left and a right. And the chromosomes, the pairs aren't identical because you get one from your egg donor, sometimes called mama, and one from your sperm donor, sometimes called papa. So and the different pairs are different types of chromosomes, and they do different things. Just like you can have different types of shoes, you can have a pair of, we're in Texas, so cowboy boots and flip-flops, because Texas, and sneakers, and I don't know, pumps, peep toes, soccer shoes. I'm out. I'm not a shoe person, but so imagine you have 23 pairs of shoes, and each pair you got one of the shoes from your mama and one from your papa. So that's roughly how your chromosomes work. DNA is your genetic code, and the genes are the functional units of DNA. Each one makes a protein. Although, just like any recipe, you can alter it a little bit. If you've got a good pancake recipe, then you can use that pancake recipe to make, banana pancakes or cinnamon pancakes, apple cinnamon pancakes, ooh, cranberry pancakes. And it's just a bit of editing that can turn each gene into a slightly different protein. So we're on page 23, top of page 23 of the note set. And the first word here is genotype. So we're filling the blank. Genotype is to genetic makeup as phenotype is to observable traits. And these terms will be very important on the test to know the difference between the genotype and the phenotype. So and we'll come back to that. The characters are your observable features like hair, eye, skin color, whether or not you're lactose intolerant. And the traits will be the variance in those characters. So hair color could be brown, it could be blonde, it could be black, it could be red, or some combination thereof. Just like there are different traits for eye color, brown, blue, green, hazel. Your genotype is derived from your parents' genotypes. Each somatic cell contains 23 pairs of chromosomes, and the 2 chromosomes that match up to make each pair are called homologous chromosomes. Let's see, I think I can work out how to highlight along. So oh, no, I need to make that more opaque. Genotype? Phenotype. Okay. So the homologous chromosomes are the 2 chromosomes that go together, just like your 2 cowboy boots go together to make a pair of cowboy boots. Next term, the first 22 pairs are autosomal chromosomes. And these, soma again means body, so these are your body chromosomes. And the 23rd pair are the sex chromosomes. The karyotype is the entire set of chromosomes, and we have a picture of the karyotype here. And the alleles are the forms of the gene. So you could have the allele for brown hair, red hair. You could have the allele for dark skin, light skin, somewhere in between. In each autosomal pair, one chromosome is from mama and one is from papa, and they code for the same traits. So in each autosomal pair, you get from mama a hair color gene, from papa a hair color gene, from mama an eye color gene, from papa an eye color gene, from mama a skin color gene, from papa a skin color gene, from mama and papa each genes for height, whether or not you have freckles, whether or not you have dimples, whether or not you have that chin dimple thing. Next, fill in the blank. Alleles are different forms of genes for the same traits. For example, the trait of eye color has many different alleles, but we can simplify and use a brown-eyed allele, and this will be a capital B for brown eye, and a blue-eyed allele, this will be a lowercase b for blue eyes. So if both parents give you identical traits, identical alleles, say you get, let's make this darker. Okay, sorry, playing with the color. There we go. So capital B for brown and lowercase b for blue. So in this case, if you get two of the same, B for brown, or lowercase b for blue, then you are homozygous because homo means same, you have two identical alleles. If you have, so the question on the note set is with the capital B genotype, what is the phenotype? And the phenotype is brown eyes. Next question, if the phenotype is blue eye color, what genotype is being expressed? And that'll be the lowercase b. So here again, lowercase b is your, these are your genotypes. And then the brown versus blue, the trait that it is expressed is the phenotype. So the letters are the genotype and how it shows up on you is the phenotype. If you get two different alleles, so a big B and a little b for a genotype, then you are heterozygous for that trait. Hetero means different. So because brown eye color is dominant, strictly dominant over blue eye color, if you have big B, little b, you will have brown hair. I'm sorry, we're doing eye color, brown eye color. Simple inheritance is a single pair of alleles. Polygenic inheritance is when several different genes work together. Eye color, hair color, skin color, all of these things that we're talking about as the easily seen traits are actually polygenic inheritance, and that's how we end up with so many different eye colors and hair colors and skin colors. I think we're ready for the next slide. And page 24 on our note set. Different ways that the heterozygous alleles can interact. We have strict dominance, which we just talked about in eye color, like BB. The dominant allele is expressed in the phenotype, and the recessive allele is not expressed in the phenotype. So if you have a heterozygous brown-eyed allele and a blue-eyed allele, brown is strictly dominant over blue. So if you have this heterozygous situation, then you will have brown eyes. The recessive allele is not expressed. The recessive allele is only expressed if you only have blue-eyed genes. So you would have to get a blue-eyed gene from mama and a blue-eyed gene from papa. Okay. Other things that are dominant, freckles are dominant. If you get a freckled gene, you will have freckles. Curly hair is dominant. If you get a curly hair gene, you will have curly hair. Codominance is when heterozygous exhibits both traits. So, for example, the A-B blood type. If you get the A blood type gene, you make the A surface antigens on your red blood cells. If you get the B blood type gene, you make B surface antigens on the surface of your erythrocytes. And if you get both of those, then you make both surface antigens. I'm sorry, so that is codominant. My note said it's in a different order than my PowerPoint. Incomplete dominance is when heterozygous makes a completely different third phenotype. We don't see that often in humans. The only real example that we have in the book is sickle cell anemia. Let's say we did this with colors for tulips. So let's say that red is dominant over white. With strict dominance, with a red gene and a white gene, then you would have a red tulip. With codominance, you would have a pink tulip, and with incomplete dominance, let's say you would have a striped tulip. Technically, the stripes in tulips are actually a virus, not codominance, and not incomplete dominance, but you know. Okay, predicting simple, and teratogens are things that cause birth defects. And that's further in the note set on the bottom of page 25. So while eye color is actually determined by polygenic inheritance influenced by a number of genes, we can use the fact that brown eye color shows strict dominance over blue to learn how to use a Punnett square. In a Punnett square, there you go, Punnett square. Punnett squares are what biologists do on first dates. Punnett squares, mama's genotype is always shown across the top, and papa's genotype is always shown down the side. The parents' alleles, so this is the P generation for parents, are combined to calculate the possible genotypic and phenotypic combinations of the children, and the children are the F1 generation. When the children have children, the grandchildren then will be the F2 generation. F because the Latin word for son is filius? Filia for plural? I don't remember. Anyway, it starts with an F, so that's where the F comes from. So the genotype of a homozygous dominant individual, no, we're not there yet. Homozygous dominant individual is BB, and this will be our brown eyes. Heterozygous, let me make this a thinner line so I can write more. Heterozygous would be big B, little b, which will also show up as brown eyes because brown eyes are strictly dominant over blue. And homozygous recessive, little b for blue eyes, as we said. And the letters again are the genotype, and the traits are the phenotype. I was explaining this to my daughter once when she was a little younger a few years ago, and she said, oh, so the recessive genes are shy and they only go on stage in pairs. So the genotype only becomes a phenotype when there's two of them together. I was like, yeah, that's yeah, exactly right. Okay, kid nailed it. She nailed another one the other day too. She said, it's weird that the point of golf is to play as little golf as possible. Yes, kid, that is accurate. That is weird. Okay, so using this picture, 2916A as a guide. Show the genotypes of the children as a homozygous dominant brown-eyed father with a homozygous recessive blue-eyed mother. So let me draw a Punnett square here. And let's see, it said a homozygous dominant brown-eyed father. So homo means same, and brown is big B, and papa goes down the side. And then it says a homozygous recessive blue-eyed mother. Mother goes across the top. Blue-eyed is homozygous recessive, obviously, so little b, little b. So then what happens? So if mama has 2 copies of the blue-eyed allele, half of her eggs will get this little lowercase b, and the other half of her eggs will get this lowercase b. So we're filling in the chart. We're taking mama's eggs down and papa's sperm across. So papa here has 2 brown-eyed genes. So this half of papa's sperm will get a brown-eyed gene, and this half of papa's sperm will get a brown-eyed gene. So the genotype of the children here, so this is the P generation, and now we're at the F1 generation. The genotype is big B, little b, and the phenotype is big B, little b makes brown eyes, because brown is strictly dominant over blue. So this is the trait. The genes are the big B, little b. The trait is the brown eyes. So the question, what are the genotypes of the children? Big B, little b. What are the phenotypes? Brown eyes. And how we express this is with the statistics. So there's a 100% chance, 100% forecast of kids with the big B, little b heterozygous genotype. Okay, that's page 24. Let's look at page 25. And this picture, by the way, did the same thing, but with the lower uppercase A is the gene for normal pigmentation, and the lowercase a is the gene for albinism. And so normal skin color is anything that is not albinism. And so papa has it here. Well, I was trying to do the highlighter bit. Still not the highlighter bit. Where did I put the highlighter? Oh, okay, I must change it. Okay, highlighter bit. There we go. I like that one better. So papa here has the heterozygous dominant. So this half of his sperm give the uppercase A. This half of the sperm give the uppercase A. Mama has the recessive gene. So this half of her eggs give the lowercase a, and this half of her eggs have the lowercase a. So there's a 100% chance of heterozygous children. Okay, next page. Page 25. Using figure 2916b, which we have here, show the genotypes of the children of a heterozygous brown-eyed Punnett square, and then heterozygous brown-eyed father. So father goes down the side, and heterozygous is big B, little b. And a homozygous recessive mother. Mother goes across the top, so that's homozygous. Homo means same. Recessive will be the lowercase. So this half of papa's sperm have the brown-eyed gene. And this half of papa's sperm have the blue-eyed gene. And this half of mama's eggs have the blue-eyed gene. And this half of mama's eggs have the blue-eyed gene. So you can see what happens here Let's ask the question here. What are the statistical possibilities of the genotypes of the children? We've got a 50% chance of heterozygous and a 50% chance of homozygous recessive. So the phenotypes, we have a 50% chance of brown eyes and a 50% chance of blue eyes. Remember, the phenotype is the trait, and the genotype is the genes, the letters. Polygenic inheritance, again, is more than one gene. And the 23rd pair of chromosomes are sex chromosomes. Females have 2 X chromosomes, while males have 1 X and 1 Y chromosome. So let's show that quickly. Okay, so here's our Punnett square. And so women have XX, men have XY. So half of Papa's sperm will have X's, half of Papa's sperm will have Y's. This half of Mama's eggs will have X's, and this half of Mama's eggs will have X's. Now we're backwards, but okay. So a 50% chance of XX female and a 50% chance of XY male. That was supposed to be Y's, but okay. Messy, but works. Okay, as we've said before, this doesn't always work out this easily. We said in the development that all babies start out female and they don't turn male. They don't grow the male primary and secondary sex characteristics until after they're acted upon by testosterone. And if the body, for any reason, does not make testosterone or makes testosterone fine, but doesn't have testosterone receptors on the cells, or the receptors don't work right, then the baby can be genetically XY male, but physically completely appearing female. And so this is one of the many situations that are called intersex. Intersex is in between male and female. And so they say the incidence of intersex is about the same worldwide as the incidence of redheads. So if you've ever seen redheads, you've seen people who are intersex. And some people don't know. A kid growing up with this specific type of intersex would not know until at age 16 they had not started to menstruate. And then they would go to the doctor and start to ask questions. So, plus there's another issue, is that the gene that actually causes the the gene on the Y that causes the production of the testosterone and the male can actually, as you see on the picture here with the crossing over and the recombination, can actually jump onto an X. So you can be, XX, like genetically female, but have the male gene and be male. And you can be XY, genetically male, except if that gene has been snipped off the end, then you're completely female, except that you have a Y. But without the gene that makes male, even though you have the chromosome for male. Anyway, it's all quite complex, and there are a lot of variations and permutations. Our society wants to be able to say there is male and there is female and there is nothing in between, which would be simpler. But reality tells us that that's simply not the case. That, and even hormonally, men have testosterone and women have estrogen, but there are men with higher levels of estrogen than the average woman. There are women with higher levels of testosterone than the average male. Just like there are women that are taller than the average male, like Gwendolyn Christie, who is Brienne of Tarth. She's really quite tall. So there is a wide variation of things, and the sooner our society acknowledges the wide variation between male and female and stops being so uptight about it, the better off everyone will be. Diversity is always a good thing. So these sex-linked chromosomes are chromosomes that are only found on the X. So the 23rd pair of chromosomes, back on the note set, middle of page 25, are the fill-in-the-blank sex chromosomes. Females have 2 X chromosomes, XX, while males have 1 X and 1 Y chromosomes, XY, generally. X-linked, also called sex-linked chromosomes, are genes that are only found on the X. There are also Y-linked chromosomes, and these will be things like the hair that grows out of the ears and those eyebrows. You know those eyebrows, like the character eyebrows that like older men sometimes have? Like, that's just on the Y chromosome, so it's from father to son to son to son to son to son. So why are you not an exact mixture of your parents' traits? You are half of each of their genomes. You are not half of each of their phenomes. So it's not a mix of brown eyes and blue eyes. It's a mix of a brown-eyed trait, but maybe they're heterozygous and they have a brown-eyed allele and a blue-eyed allele. So if you get the blue-eyed allele from the brown-eyed parent, and then of course the blue-eyed parent only has blue-eyed alleles, then you can still be blue-eyed. Teratogens, as we mentioned, are things that cause birth defects. There are lots of them. Avoid them when you're pregnant. The Human Genome Project was way back in 2003. This is when you get a read of the full set of DNA. And now for genetic problems. Oh, here's our map of the human chromosomes, which are so cool. And Punnett squares. So let's do the genetic problems starting on page 26. We will calculate the statistical probability that the children will be male or female. We already did that. So XXXY, probability is 50% male, 50% female. What determines whether the baby is male or female? It is the sperm. So that's what determines. So it's Papa who determines whether baby is male or female. Who gets in trouble, who, historically speaking, has gotten in trouble for having girl children? The mama. In fact, women have been killed for having girl children, and it's entirely out of our control. And what says women are worthless than men being mad about only having female children? Yeah, that's sexism straight up. So next, is it possible for 2 brown-eyed parents to have a blue-eyed child? So I want to show you how to work the Punnett squares like a logic puzzle. So what we know here is we have 2 brown-eyed parents. So we know we have a brown-eyed gene. We don't know about this, the second allele. So let's fill in what we know here. We've got this half of the eggs will have brown-eyed genes. This half of the sperm will have brown-eyed genes. Now, is there a case in which we could have blue-eyed genes? Well, what if mama has another? Nope, then all kids have blue eyes, so that won't work. What if Papa has a second brown-eyed gene? Then all the kids have brown-eyed genes, and since brown eyes are strictly dominant over blue eyes, all the kids will have brown eyes, so that won't work. What if Mama has a blue-eyed gene? What if Mama is heterozygous? Okay, well that's possible because this kid here has a possible chance of being blue-eyed. And what if Papa is also heterozygous and carrying that hidden blue-eyed gene? Then this half of his sperm will have blue-eyed genes. And voila, et voila, Also, voila is spelled like viola because it's French. Voila and viola are spelled the same. So there's no W, it's French, and it's supposed to be weird. So there you go. Is it possible? Yes. So is it possible for two brown-eyed parents to have a blue-eyed child? Yes, if both are heterozygous. What would the genotypes of the parents be? It would be big B, little b. And what are the statistical possibilities of the children? Well, we've got a 1 in 4, which is a 25% chance. Let's see, we're looking for the children's genotypes. So we've got a 1 in 4 chance of big B, a 50% chance, which is a 2 in 4 chance of big B, little b, and a 1 in 4 chance, which is 25% chance of homozygous recessive. So 1/4, 25% homozygous dominant, 1/2 or 50% chance heterozygous, and 1/4 or 25% chance homozygous recessive. Because it asked for the genotypes on this one, right? It didn't ask for the phenotypes. If it had asked for the phenotypes, then we would say there's a 75% chance of brown eyes and a 25% chance of blue eyes. So that's how that works. And then I'm going to undo all of this and use this page again for the next question. Not sure how far we go here. Okay, that was number 2 in the note set. Number 3, is it possible for two blue-eyed parents? So what's that tell you? Blue eyes is only homozygous recessive. So they're all little b's, no question there. The only question is, what do the kids end up? Is it possible for two blue-eyed parents to have a brown-eyed child? This half the eggs have a little b, this half the sperm have a little b, and no, it is not possible for two blue-eyed parents to have a brown-eyed child. Something happened, but this is not what happened. So we've got 100% blue if we're looking for phenotype, little b if we're looking for genotype, because the genotypes are the letters. Okay, delete, 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 delete. Excellent. Number 4. Okay, we haven't had this conversation and I don't see it. There are some practice Punnett squares on page 30, so flip to that. Is it possible for two brown-eyed parents to have a blue-eyed child? Oh, we just did that one. Never mind. So number 4, normal, back to page 26. I was wrong. Normal skin color A is strictly dominant over albinism. Cross an albino papa, it's recessive, so we'll make it lowercase, with a heterozygous, uppercase A, lowercase a, mama. So this half of mama's eggs, and we usually put the dominant first, the dominant letter first, and then the recessive letter, but it doesn't matter. And then papa's sperm all have lowercase a, lowercase a. So next page are the questions, page 27. What are the genotypes with statistical probabilities for the children? So heterozygous is 2 of 4. 2 divided by 4 is 0.5. So times 100 is 50%. So 100% AA, and then half, which is 50%, lowercase a. So those are the genotypes. What are the phenotypes with statistical probabilities of the children? So that's the genotype, the phenotype. Let me get PH because P is for the parent generation and F1 is for the child generation. And remember, the grandchildren are F2. So the phenotype here is 50% albino and 50% normal. And no, I cannot write the word normal without quotes. You know that. Normal skin color is any skin color that is not albinism. So, and let me undo these so we can do the next question. There we go. Or if it'd be faster to erase. Oh, it would be faster to erase. Okay, we can do that. Oh, that's not erasing. Erasing. Every time I let go, it switches from erase to draw. I didn't know it did that. That's fun. I'm being entertained by my program here. I'm using a Viddle Pro here, if you're curious, as my digital whiteboard. Okay, so we're on number, was that number 5? Nah, it was 4. 5. A mama with albinism. Mama with albinism. And a papa with normal skin color. Oh, no. Normal skin color. So we know he's got one normal gene, but we don't know about the other gene. And they have 8 kids with normal skin color. So here's what we know. And question mark, A, question mark, A. Now, if Papa has a hidden recessive gene for albinism, would it be possible for these kids to have normal skin color? No, no, it would not. So that means that Papa is homozygous dominant and Mama is homozygous recessive. Now, in the parlance of our test questions, parlance being the words, if it says that the 8 kids all have the same phenotype, then we're assuming that it's all the same. So, is there statistically a chance that you could have 8 kids with none of them being albinism in this situation? It is remotely possible, but it's unlikely. So we're going with, if the book says all the same for 8 kids, it's all the same. Did we do the stats for that? Oops. Okay. So, oh yeah, kids have big A, little a, sorry. When the children are grown, so these are the same children, what genotypes would their mates produce albino children? So those are these, we're going to say it's mama, the same kids. So this is the F1 generation from the previous problem. So what genotypes of their mates, so we're at question mark, could produce albino children? So let's fill in what we know. And then to produce an albino child, what if we had a papa with normal skin color? Now that does not produce an albino child. So what if we had a papa who was also heterozygous for the trait like the mama? Let's fill that in and see what happens. Ah, there we go. We have a child with albinism. So that is one option. Is there another way to possibly have albino children with a heterozygous mama? What if, is the only one we haven't tried yet, Papa has albinism? Then yes, it goes from a chance of 25% chance of children with albinism to a 50% chance of children with albinism. Okay, I got a notification and it paused the video, but I think I can stitch them together. It like stopped the video entirely, so but it kept the same place. So I think I can stitch them together into one video. We'll find out. Okay, so what were we doing? I think I was undoing things. Yep. Okay, so the kids that we found here, that was the F2 generation, just so we're clear. No? There we go. Okay. So we're at number 6. Color blindness is a recessive sex-linked trait on the X chromosome. So let's look at this picture from the book. And let's show a cross between a normal color vision papa with a carrier mama. So a carrier mama is a mama who has the gene as a recessive gene, but does not have the condition. Okay, so let's draw a Punnett square. Nah, let's draw a bigger Punnett square. Whoa, hello. Okay, that was weird. Sure. So let's see, we said we have a a normal color vision papa. So papa has an X and the normal color vision is a capital C. And of course the color vision gene is not on the Y, it's only on the X. So papa only has one copy of this allele. And then we said we have a carrier mama. So this mama will have an X with an uppercase normal color vision C and an X with a lowercase colorblind C, lowercase c. So here we have a daughter with normal color vision, homozygous dominant. And then here we have a daughter with a normal color vision gene from papa and a colorblind gene from mama. So like mama, this daughter will be a carrier. And then we have a son with a normal color vision gene and a Y, because the color vision gene isn't on the Y, it's X-linked. And then a son with the colorblind X and a Y. So the possible genotypes of the children are shown. They've got 4 different possible genotypes. And the possible phenotypes are the normal female, 25% chance, 25% chance of a carrier female, 25% chance of a normal visioned male, and a 25% chance of a colorblind male. That was page 27. Let's look at page 28. Okay, what are the parental genotypes that could produce a colorblind female in the F1 generation? So we're looking at female, so we're not concerned about here. And we want a colorblind female, and let's go back. Okay, so a colorblind female, so we need 2 X's with lowercase c's. So how do we arrange this? So we know we have a Y here, and papa's sperm would have to be, so papa would have to be colorblind in order for the daughter to be colorblind. And mama, for this daughter, has to be colorblind. For this daughter, so mama could be a carrier, and we could have a carrier daughter and a colorblind daughter. So what parental genotypes could produce a colorblind female? You have to have a colorblind papa, and mama has to be at least a carrier of the colorblind gene. Now, what if mama were also colorblind? Let's make that lowercase c now. Then all of the daughters will be colorblind, and of course all of the sons will also be colorblind if both mama and papa are colorblind, because they really only have that recessive trait. So next question, could a colorblind female have a brother with normal color vision? Okay, well, we just cleared not if mama is colorblind. But let's go back to the situation where mama is a carrier of the colorblind trait. So that would give us a daughter who is a carrier. And then, let's see, this son would be colorblind. And then this son would have normal color vision. So it is possible for a colorblind daughter to have a brother with normal color vision. The incidence of boys with color blindness is about 10%, and the incidence of girls with color blindness, well, men and women, is less than 1%. So it's a fun trick of how it works out for it to be possible for a colorblind woman to have a normal color vision brother. So, could a colorblind female have a brother with normal color vision? Yes. And could the colorblind daughter, question number 8, so let me erase these. So the colorblind daughter, so we're doing things with the F1 generation now. The colorblind daughter, so X lowercase c, X lowercase c. Could the colorblind daughter have sons, F2 generation, with normal color vision? And I think we already covered this in our other example. No. Colorblind mamas cannot have sons with normal color vision because Papa is giving the sons the Y, and the Y does not carry that trait. That's why this is an X-linked recessive trait. Could the colorblind daughter have daughters with normal color vision? Well, clearly Papa would have to have normal color vision. And so, yes, if Papa has normal color vision, then the daughters will be carriers of the colorblind trait and not colorblind themselves. Okay, and remember these kids here are the F2 generation because this is the F1 generation from the previous puzzle. Punnett square. I think of them as logic puzzles. I do Punnett squares because they're fun. Because I'm like that. Hemophilia is a blood clotting disorder, which is a recessive sex-linked trait to normal parents. So it's sex-linked. Normal is going to be uppercase H, and we don't know about here. And normal is going to be uppercase H, and of course Papa's a Y. So let's fill in what we know for two normal parents. So we've got a normal daughter who is not hemophiliac. We've got a normal son who does not have hemophilia. We've got a one normal gene here, and of course the Y here. But they have a son with hemophilia. So clearly the X has to have a lowercase h on it. We have to have the hemophilia gene from somewhere. It didn't come from Papa. It came from Mama. So hemophilia, a blood clotting disorder, is recessive sex-linked trait. Two normal parents have a son with hemophilia. What are the parents' genotypes? The mama is heterozygous, and the papa is has the normal trait. So list the possible genotypes of future children. So we have 25% chance of a normal daughter, 25 or 20, wait, we're doing genotypes, 25% chance of X capital H, 25% chance of X capital H, X lowercase h, 25% chance of X capital HY, and a 25% chance of X capital H, X lowercase h, Y. And then possible phenotypes, we've got a 25% chance of normal female, 25% chance of carrier female, 25% chance of normal male, and 25% chance of male with hemophilia. Turning the page. Okay, and we're on page 29, which is the last page of the last note set. Normally this goes so fast, but with the switching to online, it feels longer than usual. Multiple alleles. So A, B, O blood types have 3 possible alleles, A, B, and O. Alleles A and B are strictly dominant over O, while B and A are co-dominant. For these problems, show all genotypes. So let's list first all of the genotypes for type A. Okay, but not as highlight. Let's do them as solid. Okay, A can be AA or AO. Blood type B can be homozygous or heterozygous. And then AB can only be AB. And O, blood type O is OO. And that's recessive. O is recessive, A and B are codominant, and then A is strictly dominant over O, B is strictly dominant over O. So we have a mama with blood type A. So we've got a mama with blood type A, but we don't know if she's AA or AO, so we've got A question mark. And we have a papa with blood type B. So we've got a B, and then we've got a question mark because we don't know if he's BB or BO. And they have 4 kids with 4 different ABO blood types. So how is that possible? So we know we've got A here, and A here, B here, and B here. So what if mama was AA? Well, no, then we'd have 2 ABs, and we just said 4 different blood types. So if this isn't AA, then let's try AO. Okay, so there's an AB blood type and a B blood type. Okay, that works. And let's see, what if papa is heterozygous? Again, that gives us 2 children with the same blood type, which does not fit our puzzle. So let's go back. And the only option other than capital B for B blood type is capital BO. So papa has BO. So here's an A blood type. So that's a different blood type. So we've got an AB blood type, a B blood type, an A blood type, and then this leaves us with our 4th kid with blood type O. So if both parents are heterozygous, AO and BO, then it's possible to have 4 different kids with 4 different blood types. So fun. And let's erase all of this. Leave our blood types on the top. Okay, question 11. 2 parents with blood type A. So we know the A, and then we have a question mark. A question mark. They have 4 children. It paused again. So we have 2, we're on number 11 on page 29, 2 parents with blood type A, which we've written in. So let's follow that logically. That's what we've got. Oh, wait, no. There, that's what we've got. And we have 3 children with blood type A and 1 child with blood type O. So what if mama is heterozygous? Well, then we don't have a type O kid, so that doesn't fit our puzzle. So let's make mom heterozygous. Okay, that's possible. And what if, oh, papa, she can't. If papa was an AB, then they wouldn't have said that papa had an A blood type. They would have said papa had an AB blood type. So the B doesn't work. Lost my O. Let's try the A, heterozygous for A. Now that gives us an AO, so that's all 4 type A kids, so that doesn't work. So let's try heterozygous for type A, and then we do have a chance of a type O kid. So is it possible for 2 type A parents to have a type O child? Yes, if they are both heterozygous for type A. Next question. Almost. There we go. Number 12. Papa is blood type AB. Okay. AB. That's what we know. And Mama is blood type B. So we've got a B and a question mark. So let's fill these in. Okay, so we have a kid here with type AB and a kid with type B. And that's what we know. So let's work out what the question is wanting. They have 3 kids with type A, AB, and B. So 3 kids with type A, AB, and B. So let's try. Papa is heterozygous for B, or homozygous for B, homozygous dominant. So we've got 2 kids with type AB and 2 kids with type B, but that's not what the question said. The question said at least 1 kid with type A. So homozygous didn't work. Let's try heterozygous. And there's our kid with type A. We've got 2 kids with type B and 1 with type AB. So that's how that works. Okay, and last question on the note set. Yay! Okay. Before number 13, bottom of page 29, before DNA testing, they used blood typing to help determine parentage. Well, really, you can rule people out as parents, but you couldn't prove that people were parents, but still useful. 2 sets of parents are claiming their babies were switched by the hospital. The first set of parents are both type A. And the second set of parents are both type B. Well, the babies are both type O. So is it possible to have 2 type A parents and have a type O child? And yes, of course it is, if both parents are heterozygous. Now, is it possible for 2 type B parents to have a type O child? And it is, if they are both heterozygous. Fill the rest of that in. We've got 3 type B kids and 1 type O kid. Fill the rest of this one in. A, O, O. So there's your type O kid and your type O kid. So is that possible? Yes. Yes, it is possible if all 4 parents are heterozygous for their blood types. All right. Well, that's all I have. That's all she wrote. I do want to say that I have very much appreciated all of the work that y'all have done, even though we went online. Y'all have still worked your butts off, and I have been so impressed and so utterly shocked that y'all have stayed in the game and rolled with the punches and kept on going. And I am in awe of your determination and perseverance. And I am so proud of y'all. So I just want you to know. All right. Well, we will call it there. This has been audio lecture, I think we said 7, for A&P II Lecture Test 6. Thank you, as always, for tuning in. The education must go on. Dr. J out.